Done, sir.
Okay. Himani, please go on.
Yes, sir. Yes, sir. Good morning, everyone. My name is Himani Dhabale, and as a convener, I welcome you all on the behalf of Microbiologists Society India. On 20th May 2025, we lost a renowned astrophysicist, and thus we have gathered today to pay tribute to late Dr. Jayant Vishnu Narlikar, sir. Let's dive deep into the contributions of Dr. Narlikar, sir, in life sciences. Dr. Narlikar sir was born in Kolhapur, India on 19th July 1938 into an academic family. His father, Vishnu Vasudev Narlikar, was a mathematician and theoretical physicist who were a professor and head of department at Banaras Hindu University, Varanasi. His mother, Sumati Narlikar, was a scholar of Sanskrit. His father, was V.S. Huzur Bazar, a statistician. Doctor Narlikar went to school at Central Hindu College in Varanasi. He then studied at Banaras Hindu University, where he received a Bachelor of Science degree in 1957. He continued his education at Cambridge University, where he was a member of Fitzwilliam College, as his father had been. He completed the mathematical tripods in 1959, for which he was awarded a Bachelor of Arts degree in mathematics and was a senior wrangler. Dr. Narlikar began his research career as a doctoral student in theoretical cosmology under the guidance of Fred Hoyle in Cambridge. He was awarded a Doctor of Philosophy degree in 1963. He was then a postdoctoral fellow at King's College in Cambridge and earned an additional master's degree in astronomy and astrophysics in 1964. In 1966, Hoyle established the Institute of Theoretical Astronomy in Cambridge, where Dr. Narlikar sir was a founding member of the institute while remaining a fellow at King's College. Unfortunately, a dispute with university leadership, Dr. Hoyle, led to resign in 1972, and it was decided that his institute would merge into the Institute of Astronomy, Cambridge. Dr. Narlikar left Cambridge that year, returning to India as a professor at the Tata Institute of Fundamental Research in Mumbai, where he led its theoretical astrophysics group. In 1981, Dr. Narlikar became a founding member of the World Cultural Council. In 1988, he was appointed the founding director of the Inter-University Center for Astronomy and Astrophysics in Pune. From 1994 to 1997, he was the president of the International Astronomical Union Commission for Cosmology. The National Council of Educational Research and Training appointed Dr. Narlikar as a chairperson of its committee responsible for developing textbooks in science and mathematics. Dr. Narlikar's research involved Mach's principle, quantum cosmology, and action at a distance physics. With Fred Hoyle, he proposed a conformal gravity model, now known as Hoyle-Narlikar theory, which attempted to produce an alternative theory of gravity that is consistent with Mach's principle. Dr. Narlikar collaborated with other critics of Big Bang cosmology, including Halton, R. Geoffrey Burbidge, Hoyle, and Chandra Vikram Singhe. In 1993, Dr. Narlikar worked with Vikram Singhe, Hoyle, and other collaborators on a high-altitude balloon flight that collected samples of microorganisms from the stratosphere at altitudes up to 41 kilometers. Dr. Narlikar received many national and international awards and honorary doctorates. India's second highest civilian honor, Padma Vibhushan, was awarded to him in 2004 for his research work. Prior to this, in 1965, he was conferred Padma Bhushan. He was awarded Rashtra Bhushan in 1981 by FIE Foundation. He received Maharashtra Bhushan Award for the year 2010. He was a recipient of Bhatnagar Award, MP Birla Award, and the Prix Jules Janison of the Société Astronomique de France. He was an associate of the Royal Astronomical Society of London and a fellow of the three Indian National Science Academies and the Third World Academy of Sciences. Apart from his scientific research, Dr. Narlikar was well known as a communicator of science through his books, articles, and radio and television programs. For these efforts, he was honored in 1996 by UNESCO with Kalinga Prize. He was featured on Carl Sagan's TV show Cosmos, A Personal Voyage in the late 1980s. In 1989, he received the Atmaram Award by Central Hindi Directorate. He received the Indira Gandhi Award for the Indian National Science Academy in 1990. He also served on the Physical Sciences jury for the Infosys Prize in 2009. In 2014, he received a Sahitya Akademi Award for his autobiography in Marathi, Char Nagaratle माझे Vishwa. He presided over the 94th Akhil Bharatiya Marathi Sahitya Sammelan held at Nashik in January 2021. In 1960, he won the Tyson Medal for Astronomy. During his doctoral studies in Cambridge, he won the Smith's Prize in 1962. Besides scientific papers and books and popular science literature, Dr. Narlikar wrote science fiction, novels, and short stories in English, Hindi, and Marathi. He was also the consultant for the science and mathematics textbooks of NCERT. Dr. Narlikar, while renowned for his work in astrophysics and cosmology, also had a strong interest in microbiology, particularly as it related to the possibility of extraterrestrial life. Dr. Narlikar was fascinated by the possibility of life originating outside the Earth. He believed that microorganisms could have traveled through space and potentially seed life on planets like ours. He led an experiment from 1999 to 2003 that collected air samples from the upper atmosphere to investigate the presence of microorganisms. Interesting, right? To dig deeper about this, let me introduce the speaker for today's session, Dr. Yogesh Soche sir, Director, Scan Research Trust and Honorary Scientist, NCCS, Pune, Maharashtra, India. He did his master's study in microbiology at Garware College, affiliated to the University of Pune in 1982. He did his doctoral studies at the Molecular Biophysics Unit, Indian Institute of Science, Bangalore, India in 1989. He then joined the Institute of Microbial Technology, Chandigarh, India in 1989. During the years 1993 to 94, he worked as a postdoctoral fellow at the Department of Microbiology, GBF, National Research Center for Biotechnology, Germany. He has served on the editorial boards of Current Science, Scientific Reports, and PLOS One. With over 3 decades of rich experience in studying microbial communities, Dr. Sochi Sir has worked extensively on human microbiome of the Indian population and how it is affected by lifestyle and genetics. He was the brain behind the National Center for Microbial Resource that harbors microbes from different ecosystems. His research also focused on understanding microbiome alteration in diseases like diabetes, obesity, celiac disease with the aim of developing microbe-based therapeutic solutions. In the recent past, Dr. Sochi Sir has also served as the professor of biology at Azim Premji University. Previously, he was a research scientist at National Center for Cell Science, Pune, an institute affiliated to Department of Biotechnology, Government of India. At NCCS, Dr. Shoche Sir has developed a microbial culture collection that harbors microbes from different ecosystems, specialized microbes like cyanobacteria, anoxygenic photosynthetic bacteria, anaerobes, and antibiotic resistant microbes. Dr. Narikar sir and Dr. Soche sir have collaborated on various scientific projects, particularly in exploring the possibility of extraterrestrial life and the origin of life on the Earth. Without further ado, I welcome and request our speaker for today's event, Dr. Yogesh Soche sir, to please proceed with the session. Thank you.
Yes, thank you very much for those nice words of introduction. And I am not loss of words to say, because on one hand, it's a honor to be giving this talk, but at the same time, it's a kind of sad moment uh, for the reason for this talk is being given. So, Himangi talked about a lot about Narlikar sir and his contributions. And she also made a mention of his interest in biology and contributions to biology. And that is a part which I am going to talk to you today. I was fortunate enough to interact with him on this aspect, and subsequently also we continue to interact a lot on different aspects after this astrobiology work also. So I'll share my screen now, and yeah, so yeah, so is my screen visible? Yeah, I hope my screen is visible
Yes,
now.
sir. Yes, sir. Yes, sir.
Yeah, so what I'm going to share with you today is about the astrobiology balloon experience which we did and the background of that. So, I mean, this is Dr. Naraikar, and I mean, I owe quite a lot to him. I had a close interaction with him for more than 20 years, and I learned quite a lot from both he as well as Mangalatari. He had almost 20 years of association on various aspects, and it's indeed a very touching moment to be giving this talk in his memory. So his interest in biology stems from his interest in origin of life. So we all know that there are many theories about origin of life on Earth, and they can be broadly categorized into two. One is uh, Earth-centric theory, which says that entire evolution took place on Earth. So the primitive molecules responsible for life were formed on Earth, and then they assembled on Earth, and then the first primitive form of life came on the Earth. So this is a totally Earth-centric theory. And of course, there are many variations where the people talk about where actually the first molecule came into picture. So whether the first molecule was had a terrestrial origin or marine origin, but these are minor variations. The basically this theory says that everything happened on the Earth. There is another variation of this theory which is called, we know as panspermia theory, and the panspermia theory says that life originated elsewhere and then migrated to Earth. Again, for panspermia also there are many variations. So one variation, I mean the simplest variation is that the primitive form came from somewhere else, and then the rest of the evolution took place on the Earth. Other complicated theories state that the primitive molecules or a primordial or some rudimentary cell was formed elsewhere, and then the rest of the evolution took place on Earth. And the extreme theory says that everything came from somewhere else. So there are major variations of that, but the I mean, the extreme of everything came from somewhere else, including the higher forms of life, is something which is not at all, which has no not larger acceptance. The larger acceptance is the variations where the preliminary forms of life, prokaryotes, or even the molecules that form the life came from somewhere else. So that is a theory which is more or less has some acceptance, and over the years the acceptance for this theory is increasing. I wouldn't say that the theory is completely accepted today, but at least what I have seen in last 2 decades is that acceptance to this theory has increased. Earlier there was absolutely no acceptance for this theory, but by contributions of people like Narlikar, Fred Hoyle, Chandra Vikram Singhe, there is increased acceptance of this theory. So his interest in origin of life came from his days with Sir Fred Hoyle and Professor Chandra Vikram Singhe, because he worked with Professor Fred Hoyle
आज ना
and
एक बार जैसे कि अपलोड हो जाएगा ना, ये बेटा इनकम सर्टिफिकेट भेजा है,
and
कहाँ भेजा बेटा लास्ट में?
and Chandra Vikram Singhe was, of course, his a very good friend. So it is Professor Fred Hoyle and Chandra Vikram Singhe proposed this one variation of panspermia theory, which says that the life in the form of microbial cells was introduced on the earth sometime back in 4 billion years ago, and but the theory also stated that it was not a single time event. It continues to happen even today. So theory says that the seeding of life, seeding of microbial forms of life not only happened 4 billion years ago, but it continued to happen after that, and it continues to happen today. So that is their variation of panspermia theory, and which fascinated Professor Narlikar, and he developed interest in origin of life and in this theory. So roots of his interest in microbiology and biology lie in his interaction with Professor Hoyle and Professor Chandra Vikram Singhe. And the theory states that life exists somewhere else in the universe, and as the planet passes through space, the planet picks up these seeds of life in its tail, and when the same comet, sorry, I must have used the word comet, I don't know. So the theory states that when a comet picks up the seeds of life, when it is traveling through the space, and whenever and those seeds are present in the tail of the comet, and whenever the comet comes closer to Earth, these seeds of life enter Earth's atmosphere, and slowly they settle down and seed the life. So that is their variation, which states that this seeding of microbial forms on Earth continues to happen today with comets as agents for this transport. So that is, in short, Hoyle Vikram Singhe theory of evolution of life on Earth. And what is the evidence for that? So there has been a lot of evidence in support of that, and these are some of the examples. So the one evidence comes in the form of meteorites. So a lot of people earlier had analyzed the chemical composition and internal structure of these meteorites, and they made interesting observations. So one way to look at it is that if you analyze the chemical contents, the and look for signatures, biological signatures in these meteorites, because there are many complex biological compounds like fatty acids, nucleotides, amino acids, complex carbohydrates. These are the compounds which they do have non-biological route of synthesis, yes, but that route is very complex. So, and biological route is very simple. So the hypothesis is that if one finds such compounds in meteorites, those are a kind of indirect evidence that from wherever the meteorite has come, that place is likely to contain life, because abiogenic synthesis of these compounds is theoretically possible, but almost impossible in practical terms. So that's why people look for biosignatures in these meteoroids, and many such meteoroids, complex biological molecules were found. So one such example is Murchison meteoroids, which was found in 1969, and uh, it not only had biosignatures, but when they analyzed the internal structure by scanning electron microscopy, they found all these structures which look, some filamentous structures which look similar to modern day cyanobacteria. So these are called as microfossils, and in many meteoroids, such microfossils, as well as, I mean, there are some other meteoroids where you will see these filamentous structure which look very similar to blue-green algae today. And there is also this meteorite called as LH uh, 3084. And here in this, because it was found in Allen Hills, ALH 3084, because it was found in 84 in Allen Hills regions of Antarctica. And here you will find these structures found in meteorites, which look similar to magnetic or magnetite particles in modern day magnetotactic bacteria. So, and this meteor was supposed to have come from Mars, and this was then seen as an indirect evidence of presence of life on the Mars in the form of at least magnetotactic bacteria. So this is again a microfossil. So such ultrastructural observations, along with the presence of several chemical signatures in meteoroids, that laid the ground to justify that it is indeed possible that life exists somewhere else in the space, in our universe, and possibly it could have seeded on the earth by theory of Sir Fred Hoyle and Chandra Vikram Singhe using comets as agents. And this is not, I mean, even today, there are some latest publications 2-3 years back where people had analyzed the chemical contents of some additional meteorites again, and they indeed found biological signatures there, presence of biomolecules in meteorites, leading to the confirming hypothesis that there could be life present somewhere else. So with all that background, it was proposed that with all this, what is required now is a direct demonstration that viable microbes exist within cometary material and they are being transferred to the earth. So that is the kind of evidence that we were looking for. And as we know, microbes, especially bacteria, are very hardy. They can survive extreme environments. On the earth itself, we have evidence of microbes surviving in extreme environments in the form of extremophiles. So we know that these extremophiles are present in hot springs, frozen environments like Antarctica and Himalayan glaciers. They are also present in oligotrophic environments, endoliths like endoliths and rock surfaces. These are the not only oligotrophic but very dry environments with very less water, and they can survive. There is also evidence. There are publications where the microbes are shown to survive for millions of years. There were papers where people recovered some fossils and they found live microbes in these fossils. So there was a case where in amber resin, one microbe was found, and when it was grown in the laboratory, they could grow in the laboratory. Again, there was another publication where bacillus was isolated from a salt crystal, which was something like 250 million year old. And then when grown in the laboratory, it could grow. So it was already shown that we already know that microbes are very hardy. They could survive extreme environment, and they can survive, they can remain dormant for millions of years. And after that, given a proper environment, they can survive and grow. So based on that pretext, again, the cometary seeding of microbial life, it supports, because since microbe can survive no air, no moisture, extremely low conditions, extremely low temperature conditions for a longer periods of life, it makes them very suitable agents that can be transferred in space from one place to another place. So that kind of laid the background of this theory. It supported this theory and paved the way for the experiments that were suggested by Professor Narlikar. So this is the background of what we did. And so earlier also people had tried to study presence of microbes in the space. So the theory is that then the microbes being so hardy, being able to withstand harsh conditions, they can travel through the space and they can survive for a long time that is required for a life to traverse through long distances in universe, because the distances could be several light years. And theoretically, then, if some microbial cell can survive 250 million years, then it can survive the long space travel also. So that's the reason people had earlier tried to look for presence of life in the upper layers of atmosphere for different reasons, not with this idea in mind. So way back in 1943, people had tried to look for viable microbes, but this was done at lower altitudes. It was done through aircraft and exposing the plates through aircraft window, but still at the higher altitudes, the microbes were found. The interesting experiment was done in sometime in 1975 by Russian researchers. There they used a rocket to capture the viable microbes from higher altitudes as high as 48, 58, 77 kilometers. And they used very ingenious design of having cellophane and then capturing this these spore forms in the cellophane and then collecting these capsules and trying to grow the microbes in the laboratory. So this was done way back in 1975, and they indeed found live microbes even at the altitudes as high as 77 kilometers. So there was already existing evidence that live microbial forms could be found at higher altitudes. And this led to the proposal of doing these astrobiology balloon experiments by Professor Narlikar. And since he was an astrophysicist, he had connect with places like TIFR and other places. So putting together the expertise from these places, he proposed, because TIFR that time was making smaller balloons to be sent to the higher altitudes in atmosphere to collect air samples to monitor the quality of the air and study the chemical composition of the air. So his hypothesis was something like this. So if we look at the different layers of atmosphere, the flights fly at this altitude. They fly at up to 12 kilometers. So anything coming from human interference would maximally come up to the 10 to 12 kilometers. Above that, there is no human interference. Even the dust storms and clouds can reach only up to here. but stratosphere, there is very unlikely to be any human interference. So his idea was very simple, that if we can send a balloon at higher altitudes, collect air samples from there, and try to see if one can get the live microbial forms there, because if the Hoyle Vikram Singh hypothesis to be believed, the microbial seeding continues to happen through the atmosphere. Slowly the microbes settle. So in higher layers of atmosphere, if you find the live microbial form, it is a kind of proof that microbes are being seeded by comets. So that was his hypothesis. But then that time, the TIFR had the capacity to send balloon only at the lower levels. But after this suggestion, TIFR put together the team and they developed their capacities to send balloons to higher altitudes. So they started sending balloons to higher altitudes, and then I mean this balloon was then used for doing astrobiology balloon experiments. They already had capacity to develop balloon to the stratosphere, but for chemical analysis, and these balloons were typically smaller because the payload which they carry is quite small. Here, by virtue of design, a more complex and heavier payload was required, and hence the much larger balloon was required. In a couple of minutes, I'll show you the picture of that balloon. So this was something like his idea, and as I mentioned, that the balloon here could go up to the stratospheric altitudes and collect the samples. So that was the overall idea about this particular experiment. So this study was then was done in 2005. So in which IUCA, ISRO, because ISRO was the agency which supported this research and developed the capacity to develop the payload to collect the air samples, and TIFR balloon facility designed the balloons for this purpose. The experiment was done twice actually. In 2001, one balloon was sent. That time CCMB was involved and CCMB analyzed those samples. Later on, it was thought that the experiment should be replicated in two independent labs, and that's how the NCCS came into the picture in the balloon experiments in 2005. So this is the balloon. As you see here, this is the huge balloon that was sent in April 2005 from Hyderabad, because the TIFR balloon facility is present in Hyderabad. And you can imagine the size. This is something like 26.7 million cubic feet of balloon of size, and it has 450 kilograms of scientific payload. I'll show you that payload in next slide. And it used 38 kilograms of liquid neon. So that time the liquid neon was used as a cryo suction, for cryo suction. And since this liquid neon was not available in India that time, it was imported and it was used as a cryo suction. So so this is how this is what the payload looks like. It consisted of these 16 capsules, which was designed by ISRO. It contained 16 capsules made up from high quality stainless steel and with a valve that can be opened by telemetry. So the valve can be opened from Earth by telemetry, and there were 16 such tubes present in one holder, and this payload was attached to a balloon. And the idea was that at different altitudes, by using commands from command center on Earth, the different capsules will be opened, the air will be sucked inside at that altitude, and then the valve will be closed again to avoid contamination. And out of 16, one tube would remain as a control. It will remain unopened throughout the experiment as a control to prove that it is indeed sterile. And out of remaining 15 tubes will be opened at different altitudes. So that was the overall experimental design. And after the all the sample, I mean, the air was sucked in all the tubes, the balloon, that huge balloon which you saw, that would get cut off from the payload. And the payload, of course, had a sensor. So then the payload will fall somewhere. And since it had a sensor, one can find out where it had fallen. So the vehicle will go and collect the payload and bring to the laboratory. So that was the experimental design. And then after the payload goes to the laboratory, then these tubes were taken out. And in the 2005 experiments, the tubes were distributed between two labs, NCCS and CCMB, where it was analyzed. So CCMB in 2001 had found out some bacteria in these tubes, and they had also found 3 novel species of Bacillus: Bacillus aerophilus, Stratosphericus, and altitudinus, where these 3 novel species of Bacillus were found at these stratospheric altitudes. So this was in 2005. So when the tube comes to the lab, the air is released from the, because the air is pressurized, so the it gets converted to liquid. Actually, in liquid neon, the air gets converted into liquid. So once it is brought to the lab, the air is released through a 0.2 micron, slowly released through a 0.2 micron filter, so that the any bacterial form that is present in the air could be captured on the surface of the filter. And then this filter is taken out, cut into different pieces, and grown into laboratory media to promote the growth of the organism. It is also possible that some bacteria may be adhering to the walls of the tube. So then you put phosphate buffered saline in the tube, shake it vigorously, and then again filter that saline through similar filter. And then again, whatever is captured on the surface of the filter, you try to grow that in laboratory in different media. So this was a 2005 experiment where, since it happened in Pune, NCCS, Professor Narlikar himself had come to our laboratory when the samples were being analyzed. So this is my colleague Shrikant Pawar, and who has now his own business in Pune. And Parag Vaishampayan was my research associate that time. Today, Parag, this experiment kind of helped him to make his career. Subsequently, he joined NASA, and currently he is working with NASA as a scientist. So yeah, so then as you see that these filters were cut and grown on variety of media. And in 2005 experiment, uniquely, we found hello, a bacterium growing from the filtered air. We found a bacterium growing on the on this particular plate. So this was for the first time a live bacterium was found at these altitudes in 2005, directly in the air. In 2001 experiments, the bacteria were found in washings, but in this we found it on the in the air itself. So when this was isolate was analyzed, the 6' rRNA sequence found it to be a member of genus Janibacter, but likely to be a novel species. So we did then complete chemotaxonomy of this particular isolate, and we named it on Professor Narlikar's suggestion only. we named it Janibacter hoyle to honor the original proposer of this theory, Sir Fred Hoyle. So it was named as Janibacter hoyle. So these are the scanning electron microscopic and transmission electron microscopic pictures of Janibacter hoyle. And here you can see that it's a novel species of the genus Janibacter. So this we published in International Journal of Systemic and Evolutionary Microbiology. And these are, okay, this is, yeah. And in addition to this, in the washings, we also found many other species of Stenotrophomonas, Acitrobacter, Bacillus, Janibacter, and Methylobacterium. But these are not novel species. These were already known species. We also did DNA-based analysis, and we also found some other novel species, some species, known species in the DNA-based analysis also. This organism was resistant to ultraviolet radiation, and also it was resistant to gamma radiation. So we also did genome sequencing of this organism that time, and we found that it has many pathways that make it resistant to harsh conditions of stratosphere, many metabolic pathways, very good DNA repair enzyme system that helps its survival in stratospheric environment. So these are all, I'll not go into details of these complications. In the same study, CCMB also found two new species of genus Bacillus, which they named as Bacillus israelensis and Bacillus aryabhattai. So that was overall the 2005 experiments. The question was, does it conclusively establish the extraterrestrial origin of these organisms? Actually, no. The fact that they are present at this higher altitude does not necessarily mean that they are coming from outside, because there still remains a possibility that these could be a contamination from the earth, and that possibility cannot be ruled out. So the one way to address this question is to do something called a stable isotope ratio. So typically, carbon and nitrogen exist in two isotopic forms. These are non-radioactive stable isotopes, and the ratio of these isotopes in the organism is reflection of the ratio of their origin. So in Earth, we have a certain ratio of C12, C13, N14, N15. So all the organisms will on the Earth will have the same ratio as it exists on the Earth. And the organisms coming from somewhere else would have the ratio that is indicative of that particular place. For this, it is important to carry out a single isotope ratio on a virgin organism. Virgin in the sense that once you capture the organism on the filter, that single cell should be subjected to chemical analysis. Obviously, if you bring it in the laboratory and grow in the culture medium, it will lose its original isotope ratio. So that time, these techniques were not available in 2005, but subsequently, these techniques were developed. So then it became possible to do a stable isotope ratio analysis on a single cell, single bacterial cell. And this is a technique which is now very widely used in microbial ecology. Today, even today, unfortunately, we do not, we have only one equipment in India at PRL Ahmedabad. They acquired that instrument few years back. And the techniques for using this for biological purposes are now very well established, because as I said that it is being used very extensively in microbial ecology today, and the technique has become so powerful that today it's also possible that you don't lose the cell when you carry out this ratio. So what will happen is that you have a single microbial cell, you do stable isotope analysis on that cell, but you don't lose the cell. You can take that, whatever is remaining of that cell, you can take that cell, and in the modern day molecular biology, you can amplify that DNA of that cell and then subsequently do genome sequencing. So what I want to say is that it is possible that you capture a single microbial cell in stratosphere, bring it to the laboratory, do a stable radioisotope as analysis to prove its origin, then take the same whatever is remaining, you can amplify the DNA from that single cell and do the genome sequencing of the same cell to identify what organism it is. So today the technique has developed for that, but unfortunately more than 10 years we along with Dr. Narlikar followed up this with various authorities to take this further, and looking for ISRO's support to do these experiments, but unfortunately, it could never happen for variety of reasons. It could not happen. So I think that is a sad thing that his wish could not be fulfilled during his lifetime. And today, if somebody still takes up, pursues this with ISRO, and these experiments are concluded, I think that would be a great tribute to him and his memory. And what I would like to emphasize in my concluding this thing is that these experiments, though they were done in 2005, it show that how much ahead of time he was thinking always, not only in astrophysics but also in biology and microbiology also, because similar experiments in different forms were subsequently repeated by many other researchers. And NASA believes that looking for live life forms at higher altitudes is very important. They may not be looking at it from the perspective of panspermia theory, but they say that if life can, this is the from NASA website. So if life can survive the conditions in the stratosphere, life can also survive in space. And it says that knowing how and why tough organisms persist in the stratosphere above our heads will be important when it comes to protecting planets that we explore in the short term. So point is, even if you prove that the microbes you found in stratosphere are not of extraterrestrial origin, they are from planet from Earth only, but somehow they have gone to stratosphere and survived there, that also still means quite a lot. It is a very important scientific knowledge, and in that way I would say that how he was thinking well ahead of his time. And a lot of other people carried out these experiments, as I mentioned, in variety of forms. People carried out experiments. Those results are already published. These are again some of the results where people looked at the live microbial forms in stratospheric altitudes. There are paper published in as respectable journal as ISME, where they looked at live microbial forms at stratospheric altitudes and look for reasons for survival of organisms there. So finding out the stratospheric organism and study for life in stratosphere is a new field that has emerged in last couple of years. Unfortunately, under Narikar's leadership, we made the beginning, but we could not pursue it for a variety of reasons. And again, I repeat that if somehow we could pursue this and make further forays in this field, it would be a tribute to his memory from the microbiology community of India. I think with those words, I'll stop here. Thank you very much.
Thank you so much, sir. Any questions that you might have are welcome now.
Yes, before the question, I would like to make a comment. There is a great loss. We are losing Honorable Dr. Jayant Narlikar. He will be among microbiologists because of his theory, because of his work. Today, we try to pay condolences by arranging the lecture of probability, the most perfect person who was very close to Dr. Jayant Narlikar. I'm not talking about the family relation. I'm talking about the research relation. He has told what experiments were carried out and what new theory was proposed by Dr. Jayant Narlikar. I request Himani, at the end, every one of us will stand and will pay condolences to Honorable Dr. Jayant Narlikar. One more thing again, we are preparing one resolution. We are passing one resolution on behalf of Microbiologist Society, and will send by post at his home, because his family should know that how Indians were loving this beloved scientist, beloved microbiologist. Now the session is open for discussion, question and answer. Please go ahead.
There is Kirat Kumar Ganguly who says that I have a question. What is the question?
Kirat sir, please. Kirat can ask the question. He is from West Bengal and he was the state president of Microbiologist Society last year. Kirat sir, you can ask the question.
He is not there.
I think some technical issues are there. Dr. Prakash Halami is here. Dr. Swarnajit Singh sir is here. And many of our state president from Chhattisgarh, Bihar, Tamil Nadu, and from different Gujarat, from different parts. Yes. Anyone, Dr. Rupa ma'am is there. Anyone, GV Mali is there from Maharashtra. Yugal Kishore is there from Assam.
I have one question.
Yes. Ask sir, please. Go ahead.
Yes. In this balloon experiment, from the air samples, the scientists, under the guidance of Dr. Narlikar, they have isolated bacterial species. Is there any fungal species?
Oh, yes, yes.
Right.
Fungi were also isolated. There
You are
are
not mentioned. You are not mentioned here.
fungi were isolated. I did not show the complete table, and fungi were isolated at CCMB, but there were two species of fungi were also isolated, though not novel species, but fungi were also isolated.
Can you name, please, sir, this species?
It was
Genera?
Fusarium and Penicillium.
Which one?
Penicillium.
Okay.
And sorry, not Fusarium, Aspergillus and Penicillium.
But for this fungal species, the same strategies there, they are from Earth only.
No, the hypothesis remains the same. That they could be of extraterrestrial origin, not from. This is what we would like to believe.
Okay.
Yes.
Thank you, sir.
Any other? Want to ask the question? Otherwise, we'll conclude. Yes, Kirat sir is there.
Yeah.
Kirat sir has joined. Dr. Kirat. Dr. Kirat, you can ask the question.
Yes, sir. Sir, can you hear me, sir? Can you hear me?
Yes.
Yes.
Okay.
Yeah.
Sir, I had a very fundamental question. That is, at the time I understand it was 2005. Was it able to do next generation sequencing? Because in addition to the culturable bacteria, there may have some non-culturable bacteria. Is the samples all present at that time also, at this time also?
Yeah,
So
yeah.
that,
So I made a passing remark on that. That yes, uh, I mean, 2005, though we did not have the metagenomics and these things available, but then subsequently, since we had preserved the samples, the washings, we did amplicon sequencing on that. And we again identified, we did not do shotgun metagenomics, but we did amplicon sequencing and we identified DNA sequences of some other organisms also. Like pseudomonas, and so some of the organisms which we isolated, that DNA plus some other organisms were also present, but that study was, unfortunately, we have not yet published.
okay, thank you, sir. Because the probability of getting novel species which are
Yeah,
not
yeah,
culturable will be increased in that
yeah.
way.
But that time, we, of course, that time the metagenomic shotgun
Yeah,
metagenomics was not available, but we
yeah,
did
exactly.
amplicon sequencing.
Excellent, encouraging work, sir. And
Thank
this
you.
we could be able to hear you just because of our beloved Microvalue Society,
Thank you. Thank
Dr.
you.
Deshmukh, sir. Thank you, sir.
Thank you.
Uh,
Yes, anyone or Indumati ma'am, she is praising your lecture, Indumati ma'am. But do you have any question, Indumati? She is from Chennai.
No. Sir. I was just praising for the comment initially for the lecture and now I was just supporting what Kirat sir said. That's very true.
Okay,
Many. Thank you, sir.
okay, no problem. So, now Himani ma'am, you can proceed.
Yes, sir. Thank you so much, sir. It has been such an honor to be a part of this event. On behalf of Microbiologists Society India, I would like to extend my heartfelt gratitude to the speaker of our session, Dr. Yogesh Soche, sir, Director, Scan Research Trust and Honorary Scientist, NCCS Pune. And sincere thanks to Dr. A.M. Deshmukh, sir, All India President, Microbiologist Society India, for organizing this event. Finally, thank you to all the participants who joined. Thank you to one and all. Lastly, let us take a moment to acknowledge a great loss of our nation, Dr. Narlikar, sir. Please join me in 2 minute silence as we express our condolences.
Yes, so everyone will stand
Please
at his
start.
own place. Please. Om Shanti Shanti Shanti.
Thank you, everyone.